My name is Dosia. I'm a rising junior here at Mary Wash. And this summer, I'm working with Dr. Smith in the chemistry department on water treatment. We're using the 3D printer to print our own columns for the HPLC instrument. And our goal is to be able to control the path in the column to to improve our results at the end of our water filtration. I want to say a big thank you to all the donors for making my research with Dr. Smith in water filtration possible this summer. Thank you.
Well, good afternoon, everybody, and welcome back to IDIS 300 Life After COVID. My name is Anand Rao, Professor of Communication and Chair of our Department of Communication and Digital Studies here at the University of Mary Washington. I'm really looking forward to today's presentation. You know, over the last several weeks, we've had wonderful presentations talking about the impact of COVID on many aspects of our life, and as well as looking at historical parallels. But for us to have any true understanding about life during COVID or life after COVID, we need to have an understanding of the science of COVID. And that's why today's presentation is so important. I'm excited to welcome Dr. Swati Agrawal, who is an assistant professor of biology here at UMW, for the presentation. Now, before I turn it over to her for the presentation, I want to remind you of a couple of quick things about the format, as well as the tools you have available to you during this call. We'll have about half an hour for the presentation, and then following that, I will return with Drs. Mellinger and Lewis for a discussion of the Q&A questions that you pose during the presentation. Now, you have two ways to communicate with us during the presentation. One of them is the chat box, and this is really only if you find there's a technical difficulty you need to communicate with just us panelists. You can send us something through the chat. But more importantly, you should be looking at the Q&A box, and that is for where you can post the questions that you'll have for Dr. Agrawal, and to be able to then upvote those questions. Remember, there's an upvote, a thumbs up logo that you can click on to be able to upvote those questions. And the ones that get to the rise to the very top are the ones that we will ask first when we get to the Q&A. Well, without any further ado, let me turn it over to Professor Agrawal for the discussion of the science of COVID. What's next?
Thank you, Dr. Rao. It's amazing to be here. I've learned so much listening to the past few lectures from the experts at our university. And it helps me broaden the perspective, right? Helps me understand the pandemic a little bit better. Perhaps helps me deal with some of the vaccine hesitancies that we are seeing in our world, and deal with all sorts of questions better. So today I am excited to share my presentation on the science of COVID, what's next. And so, as I said, we did one of these series last year, and we've learned so much over the course of 1 year. So what I'm going to try to do in this talk is recapitulate what we really know about the virus, the treatment, and of course, the big part of the treatment is vaccine, which is, treatment before you hopefully get the disease. And then see what lies next in terms of, again, treatment, in terms of changing social behavior, such as vaccine hesitancy, or, preparedness in terms of, newer variants that are coming out, or next pandemics that come out, and also highlight some of the things that the science that went into COVID research has done for the next set of infectious diseases that we're going to deal with, because we're not done. These sort of viruses, bacteria, pathogens are going to come time to time. And now, because the world is so hyper-connected, we can expect that many of these pathogens will cross borders like this one, and we'll have to come up with ways to deal with it. So I'm sure over the last year and a half, everyone's got to know the beautiful structure of the virus, and there's no pun intended. It is indeed a really beautiful virus. Under the microscope, it really looks like the corona of the sun because of the spike proteins that exist, that decorate, but yeah, a deadly one, right? So I want to highlight a few of the structural features of the virus that are going to make sense in context of the rest of the talk. And many of these words you might know, but what I want to try to do is highlight what is the biology or the chemistry of these structures that relate to, let's say, the kind of vaccine we receive, or the kind of treatments that we have for patients who get COVID. So it is a virus that the genome is an RNA. And this context, the RNA is very different from, when we start talking about the mRNA vaccines. But the genome is a single-stranded RNA, and it's much like the influenza virus. The surface of the virus is decorated by spike proteins. Again, we've heard a lot about the spike proteins. These proteins, the glycol part refers to a carbohydrate that is attached to the protein. This is, as you might know by now, this is the first point of contact for the virus with the host cell. This is how the virus gets attached to the host cell. You might have heard about the ACE2 receptors, and the spike protein helps it attach. Now, because this is the point of contact, you can expect that the protein really is important, and that's what our vaccines are currently targeting, rightly so, because again, that is the way the virus makes its entry into the host cell, but also it's rapidly mutating, and we need to be aware of what those mutations are and what it means in terms of the disease outcomes. A few more structural details. So the spike protein is embedded in this nucleocapsid, and then there is also a envelope. And the envelope, the best way to describe the envelope is sort of like an oily layer, a hydrophobic oily layer that the virus actually steals from the host. And so what that means for the virus is that the virus is able to get entry into the host easily, because the envelope looks much like the host cell, the outer surface of the host, and so that therefore it is easily able to get into the host, because it's once upon a time it stole it from the host. Okay, so we now know that at some point these viruses originated from bats. Bats are known to have many coronaviruses. It's amazing if you read up the literature, the number of coronaviruses that bats can have at any given point. But there was possibly an intermediate host. I think at one point we know that it also went through pangolins, but we really don't know what was the most recent host before the virus moved to humans. And we call these viruses zoonotic viruses, which move from animals to humans. Okay, so in the past few years, and really amazingly very soon after the virus was discovered, the structure of the spike protein and the protease of SARS-CoV-2 was resolved. And this was a very big step in the development of drugs and vaccines. So unless you know what you want to target and the structures and nitty-gritties of it, you will not be able to develop a vaccine or a drug. And this happened pretty soon as around January of last year, so January 2020. So a little bit about the disease and what we've known about COVID-19. Again, up until last year, there were lots of unknown. What kind of disease does the virus cause? How easily is it transmissible? And that aspect is still changing. But what we know is that the virus causes a pneumonia-like disease, much like what you would experience with flu. It can result in ARDS, which is the acute respiratory distress syndrome, which means that patients have really trouble breathing. Their lungs feel like they're drowned because they're not able to function. So that's one aspect of it. And pretty soon, when we started to see this, we also to see something else, which is that in some cases, and in mostly younger individuals, we saw something known as cytokine storm. And the closest analogy to cytokine storm would be your allergies that you experience when spring comes around. And so this is basically a hyperactive immune system, which is trying to control the virus, but in doing so, it ends up harming the tissues of the lung itself. So really quickly, the anatomy of the lungs is that the air goes in through the trachea, and then it branches off into these bronchioles, and then each of these bronchioles puff up into these little air sacs that are called as alveoli. And in a normal alveoli, the outer layer of the alveoli is thin enough so that there can be gas exchange. So there is exchange of oxygen into the blood from the air that you've just inhaled, and the carbon dioxide from the tissues gets back into the alveoli so that you can exhale it out. Now, you can imagine that if these alveoli are not able to do that, then it can be pretty disastrous, because our tissues need oxygen to burn the food, metabolize everything that we do. The food that we eat needs to be broken down with the help of oxygen. So if oxygen is not getting to our tissues, to our organs, then it can have a severe complication. So how does that come to what the virus does? Well, I've told you earlier that the virus binds to the ACE2 receptors, and the ACE2 receptors are present in your respiratory tissues, such as the linings of your nose, the nasal lining, what we call as the epithelial tissue. And so by binding that, they get entry into the lung, and then they attack these alveolar cells so that they're not able to perform the function. They're not able to do the gas exchange. And then for the cytokine storm, once the body recognizes that it has the virus, what it does is it will mount an immune response, and it will release what we call as cytokines, which are basically chemical molecules that signal the army or the various cells that can attack the virus. And that's good, except that it overdoes it, and especially in individuals who are younger and have a active immune system. So we've talked a lot about people who have underlying conditions such as hypertension and diabetes that are particularly at risk because they're, in some ways, they have compromised immune function, but too much of it can be bad. And the way it is bad is because, as I said, the cytokines signal a bunch of cells to infiltrate the lung tissues, and they can end up causing clot formation, scar tissues, inflammation, and all of those are also bad in helping the alveoli perform its function. So what I want you to take away from this slide is that we know it's, and I have heard many podcasts, many talks by physicians who have said that this really, in the hospital, they have started looking at this like a flu-like disease. Like, they've started treating the symptoms of pneumonia, of the acute respiratory distress, and trying to control the cytokine storm. Which brings me to my next slide, which is what did we learn about the treatment, and what do we know now? And again, going back to last year, our hospital systems were getting overwhelmed because we really didn't know what would work. What we've learned is that there are 3 ways in which we can approach the virus. And although treatments have been less successful than vaccines themselves, what we know is that there are 3 ways to sort of attack the viral replication and the cytokine storm. The viral replication it can be stopped by inhibiting some process in which the virus multiplies. And so there are drugs that will inhibit that enzyme that is going to help the virus make new RNA, that RNA that you saw in the first slide. And so there can be antivirals that can be used to that end. And then there are also some other drugs like remdesivir, which we call as nucleotide analogs. And what nucleotide analogs are, imagine the virus genome, right? And it's made of the 4 letters. So you've heard that DNA is made of 4 letters, probably A, T, G, and C. RNA is made up of 4 letters, A, U, G, and C. And the nucleotides is each of those letters. And it's the specific arrangement of those letters that give rise to one kind of mRNA. Now, remdesivir looks like one of those letters, except that if it is incorporated in the mRNA, then the mRNA cannot make a copy of itself. And so we've seen some data, although there is a lot of, there is a little bit of lack of clarity, and mostly this is because of the fact that the disease is not the same. It's not the same in different parts of the world, with different variants, with different age groups, with severity. So we really don't know, if remdesivir works in every case, but it has been used extensively in this past year to treat severe COVID patients, hospitalized patients. Anti-inflammatories, again, to control that cytokine storm. And so dexamethasone was actually recommended by WHO on September 2nd, 2020. Dexamethasone was recommended for treatment of patients with non-severe COVID-19. And some other corticosteroids are also have been recommended, have been used successfully in this category. Again, the timing of the treatment is important. And for dexamethasone or for these corticosteroids, it is when the patients are not too severe. That's something that they have noted. But they've narrowed down the dosage of dexamethasone, and it's pretty commonly used now. Antibody treatment, so you might have also heard about this. I remember beginning of last year, there was a lot of talk about convalescent plasma, and that has kind of dimmed out. But what has really emerged is monoclonal antibody treatment. And so I want to explain this just a little bit so that you can understand it in context of the variants and the different variations of the viruses that are coming forward. And what that is that you grow cells and in the lab, and then you infect with coronaviruses, and the cells will produce antibodies, and those antibodies are then administered to patients. And the idea is that because these antibodies have already seen the virus, they can attack the virus. So if you're giving the body ready-made antibodies, then at least some of the virus can be controlled. Again, I've heard from several doctors that this is again effective in the early stages of the disease, and which makes sense. The way I think about this is that the early stage of the virus need control of the virus numbers, and the later stages of the disease needs control of these inflammatory responses because the immune system is now all wacky and it's trying to attack it from every angle, but it ends up causing more harm than good. All right, so I'm going to switch gears a little bit because I do want to talk about all aspects of what is changing, and so what you've probably heard in the news a lot recently are the different variants. So I think we were towards late fall last year, we were hearing about the UK variant, the South African variant, the Delta variant. So the question is, how do people know? How do scientists know? How do they keep track of all these different variants? What do they mean when they label these variants in different categories? How do they collect data? How much data is enough? And what do we need to do with that data? Early in the pandemic, you might have heard that the United States was struggling to meet the demand to test people for SARS-CoV-2. So what that meant is that officials didn't really know how many people had COVID, and they were left to respond to pandemic without knowing how quickly it was spreading. Well, now what has happened is the US faces a similar issue with a different type of test, and that test is genetic sequencing. So this is not just knowing whether you have the virus or not, it is knowing whether you have a specific variant or not. Do you have the original Wuhan virus or the UK strain, the South African, or the Delta variant? And how do you get to it? Basically, the antigen test, the PCR test that we were all, I guess getting at some point was looking for one of the sections of the virus, specifically the nucleocapsid protein. Well, the genome sequencing reads all the letters in the mRNA of the virus. And then you can compare the different strains of virus, different variants of virus that are circulating globally. So you can imagine that we need to do this at a very large scale. And even though in the past year, we've sequenced more than 360,000 SARS-CoV-2 genome, we're not there yet. If you look at the proportion of the number of genomes that US has shared on this platform, which is basically a public platform that allows free public access to these genomes, it is less than 0.3% of its total number of COVID-19 infection. So as compared to some other countries like the UK, which has contributed 5% of the genomes, 12% in Denmark, and almost 60% of the genomes from Australia. And so just a little bit on how we can make this process easier and more effective. Even now, for various reasons, if you're trying to travel globally, or if you think you have the COVID, you go and get tested, right? The testing centers already have your samples, but they do not have the capacity to do this genome sequencing. It has to go to a special facility, either nationally or internationally. And the testing centers don't have proper guidelines as to how to store these samples, how to process these samples. They don't even have the permission to store these samples in some cases. So that's such a big miss. We have the samples, but we we throw them away. And so if we could have a way in which the testing centers can regularly, I guess, transport and send the sequences to a place where they can be sequenced, we would be collecting more data and doing much more genome surveillance than we are doing currently. So what are some of the variants of concern? I'm sure you've heard about all of these variants recently in the news. So I'm going to talk about how they get the names, how WHO came up with this labeling system, and what does it mean when WHO calls a variant of concern. And so WHO, in collaboration with partners, expert networks, national authorities, institutions, and researchers, have been monitoring and assessing the evolutions of SARS-CoV-2 since January 2020. And so during the late 2020, the emergence of variants that posed an increased risk to global public health prompted the characterization of specific variants of interest and specific variants of concern in order to prioritize global monitoring and research, and ultimately to inform the ongoing response to COVID-19 pandemic. And at the bottom of this, these different variants are different from each other in having that different mRNA sequence, and specifically on the spike protein. Most of these mutations are present in the spike protein. And so what we've seen is that these are few of the variants that are called variants of concern that have shown increase in transmissibility or detrimental change in COVID-19 epidemiology, or they have increased virulence in or disease outcome, or they have a decrease in effectiveness of public health and social measures, or available diagnostic vaccines and therapeutics. So for example, yes, I have this information on this next slide. So this is the Delta variant, and what you can see here are some of the mutations that Delta variant has. These 4 mutations, I'm not going to go into details, happy to talk about it if you have questions. But basically, these 4 mutations are responsible, and they have been studied in the lab, and they are responsible for either increased entry into the cell, increased replication of the virus, easier spread of the virus, where maybe single mask is not enough. 3 feet distancing is not enough. You need 6 feet of distancing, and severe outcomes of the disease. You might have also heard about this term, which is called R-naught, which is really helpful in explaining how transmissible a virus is. And there is some data that tells us that the Delta variant is highly transmissible. So if you see here, the R-naught basically tells you how many people can be infected from that original 1 person who is infected. And so the original Wuhan virus had a R-naught of 2.4 to 2.6, which was high in itself as compared to the flu virus, which is about from 1.4 to 1.8. So the higher the R-naught, the more the transmissibility. The Delta variant, well, 1 infected individual can spread it from 5 to 8 people. It's also been seen, and actually, unfortunately, I've seen this in my own family in India, because India is heavily infected, there's lots of cases of the Delta variant, that the disease outcome is more severe. The disease progression is more rapid. This is definitely a concern in countries or states that have low vaccination. They could defy one of the tests if there are mutations that do not allow detection of this virus. Then it could be that the tests that we have to detect the virus are not enough. It could be a concern for younger age groups who are not vaccinated. And then there could be a need for a new diagnostic tool to detect the new variants. All right, coming to the most stellar part of this pandemic, something that I don't think would have happened if this pandemic didn't happen, which is the speed at which we got mRNA vaccine and the amazing results that mRNA vaccines are showing. So I see I'm running a little short of time, so I'm gonna just skim over this slide, and I'm happy to answer these questions if people want me to to go into detail. So mRNA vaccine, how do they work? Basically, you have a part of the spike protein, the mRNA, the basic building blocks of the spike protein, or the information for building the spike protein that you get injected in your cells when you get the vaccine. And then we have that mRNA builds a protein. Those protein can elicit an immune response. Because this is not the whole virus, it's only a part of the virus, then your immune system is now basically trained. And in case of a natural infection, you are going to much more rapidly respond to the infection. I am going to skip over this slide and come to this slide, which is what mRNA vaccine platforms did in terms of helping us with other infectious diseases. Well, the technology is really marvelous in the way that it can be adapted. If you think about a bunch of Legos that you have in the house, or the pizza dough that you have sitting in the fridge, you can imagine that a number of recipes can come out of that pizza dough, or a number of creations can come out of the Lego, as opposed to if you bought a store-bought pizza or something like that. And so basically, it is a plug-and-play approach that is going to be helpful in responding to future pandemics, in also making the booster shots that we will soon need when in the future to deal with the COVID itself. It could mean that that vaccines for other infectious diseases that are important, such as measles, polio, can be produced faster and can be produced in greater amounts to meet the demands. Sometimes some viruses, such as the CMV virus, which is a common infectious agent that can cause neurological defect in newborn, requires that you put a vaccine that is a combination of a few things. And it's much more difficult to do it using something like protein-based vaccine versus the mRNA-based vaccine, just because it is harder to assemble 5 different proteins than 5 different pieces of mRNA. And so this is something that mRNA vaccine is going to allow to do, and there's already work underway along these lines. And then finally, the Moderna is also working on vaccines to tackle other emerging infectious diseases, such as the avian influenza, Zika virus, for which the speed of mRNA vaccine could be beneficial in future pandemics. So this is the analogy I had. Another analogy that I have, which is you can take the same cake batter and mix in different flavors, and you can produce different types of cake that can appease to different members of the household if you have a household like mine. But this is basically the plug and play approach that I mentioned about the mRNA. There are many different vaccine platforms that we've seen. The top ones are the mRNA vaccines, so the Moderna and the Pfizer vaccines. The other two are the AstraZeneca and the Janssen are basically the adenovirus vaccine, where you express a part of the COVID virus in another virus called adenovirus, and you really don't get COVID infections, but you can still ask the immune system to mount an immune response. There are some other ones, such as the inactivated vaccine that have been rolled out. So this is the most popular one. My parents in India and relatives have gotten this inactivated vaccine. This is more kind of a more traditional approach. And then lastly, another kind of vaccine, which is called the recombinant vaccine, which is that protein-based vaccine that I was talking about. So we have not been able to achieve the amount of vaccination rates that that is needed, and this is just a reminder that we still need to vaccinate a large population of the U.S., but look at the rest of the world. The truth of the vaccine rollout is you're not safe if people around you are not safe. And again, given the interconnectedness of the world, really we're not safe if some of these other countries are so far behind. And in order to achieve that, there has been, I'm going to skip this. In order to achieve this, there has been an initiative called as the kovacs initiative. And this is co-led by some of these foundations, but ultimately by WHO and UNICEF. And the idea is that the developing countries can donate, can share vaccines, and they can be distributed in the developing world so that, again, we can nip the virus before it changes so much that our vaccines are ineffective. And the 6 vaccines that are part of this program are the two mRNA vaccines, the 2 adenovirus vaccine, and the recombinant vaccine. Okay, so this will be my last slide. And then I can answer anything from the slides that I left out or any questions that you have. But our priorities to address the new variants and the lessons that we have learned is that Winston Churchill once said that do not let a crisis go to waste, right? What we've learned now should help us prepare better to end this pandemic, because I really don't think we are there yet, but also to prepare for what is coming after this pandemic. There are certain, psychological things that we'll have to deal with in the coming years. There are certain other infectious diseases that we'll see pop up because of the rampant use of antibiotics, for example. And so what can we do to address these, the new variants? Well, we should continue to push and eliminate the current SARS-CoV-2 virus. I did skip a slide. I can go back if anybody wants to see it later. But what our data is showing is that 2 doses of vaccine, and this has been tested at least on the Pfizer, BioNTech, and the AstraZeneca, is showing to have at least some efficacy and a great amount of efficacy, greater than 60% efficacy against the new variants too. And so, people who haven't gotten 1 dose of vaccine or are yet to get this second dose of vaccine, if they get both doses of vaccine, a lot of the population can be protected. We need to do more of genomic surveillance. We need to do this through global sequencing. As I said, there has to be a way in which the testing centers can perhaps save the samples, and we do not let that work go to waste. And then sharing of resources is going to allow more places to do these global sequencing. We need to be able to create a central repository of samples so that we can continuously have the samples of sera from individuals with past infection or past immunization, so that we can see which variants are escaping our vaccines, our therapeutics. I think companies are already doing this, and there's a great incentive for the pharma companies to keep investing into newer vaccines that are going to help protect us against the newer variants. But we need to make sure that these we are ahead of the game. We're always keeping track of what the new variants are, and we're vaccinating. Science can only help us so much, and the rest is to do with how we do public health, how there is administration of vaccine, and how many people actually take the vaccine. And then to ensure global access, we need to strengthen COVAX initiative, improve availability, affordability of COVID-19 vaccine. One of the biggest drawback of the mRNA vaccine, it is very costly. A $5 vaccine in developing country like India is a lot. And I think the average cost of vaccine there is about $1. And so even though it's the most effective against the newest variants, if it's not affordable, then we are not able to really get to what we're trying to do, which is end the pandemic. And then with that, I will end the talk. I know I've gone some over, so I apologize for that, but I'll take any questions. And thank you.
All right. Thank you very much, Dr. Agarwal. It's great to have your technical expertise here, and it's no longer no child left behind. It's now no country left behind, right? Let's get started with the Q&A. I'm Keith Mellinger, the Dean of the College of Arts and Sciences, and I've been monitoring the question and answer box, and there are, of course, a lot of questions that have come in. You've brought a whole new perspective to the course, which has opened up to a lot of new questions here. So the first thing I want to ask you about deals with these variants. I guess the most recent is the Delta variant that we keep hearing about in the news. And what we're seeing, or at least what we're hearing through the news, is that it's more severe, that that the virus has mutated and it's become more severe. It's affecting people in worse ways. It's highly transmittable. I guess what I want to know is this normal? Is this typical? Are flu vaccines are flu viruses like this as well, that when they mutate, they actually can get worse? Or is this unusual for this virus? What's your take on that?
Right. So when you're in the pandemic, it seems like nothing worse than this can happen, right? So I've been reading data, and what it's been showing us is that this has been mutating slower than the flu virus. But to us, how does it seem? It seems like every quarter we're getting a variant that pops up. And one of the papers made a remark, which was like, why is every variant of the virus becoming stronger in terms of transmissibility, the replication? Wasn't that our concern with the UK virus? That was the concern with the South African virus. So what I want to say is that we tired, yes, after a year and a half, but still the virus is new. It has to achieve and the more the vaccine rollout, the more natural infections prevail. At some point, the virus can only mutate so much, because the objective of any pathogen, and this is really fantastic, for me, being someone interested in infectious diseases, is not to make the host die. It's to pass on to the next host. And so by increasing in transmissibility in its replication, it is compromising some of its characteristics in being able to live for or exist for a longer time. And so I guess I'm defying your question, but I guess we're really in the nascent state of the pandemic, where we are still seeing mutations, and eventually it will plateau out, and this will be a result of how much vaccines coverage we can have, how much herd immunity we have, and we can only look forward to when that day comes. But I know we are very anxious at the moment.
So is it hard to compare this to something like the flu, which we've studied so much more, just because it's so new?
Definitely. And I think one of the things that we've seen is coronavirus is a bigger family of viruses than the flu itself. That's something that I always think of is, MERS came and went. The original SARS came and went. Why is this so much worse? And why is this so much more successful than the flu virus? If you think about the severity of the disease, it's still less severe than something like flu, but it's so much more transmissible that if 100 people get it, 10 of those people that are getting very sick, the outcome is very similar, or at least equal, or at least tiny bit more than what the outcome would be with flu. And there are lots of ways to model what the next mutation is going to be, but really there is so many permutation combinations that I think the way to move forward is not to expect the next mutation and see what happens, not to throw everything at the wall and just hope that something sticks, but do what we can and what has been shown to be effective, which is our mask, social distancing, vaccine. I still think we need that. I mean, for states that do not have enough vaccine coverage, we cannot just take off our mask, right? The CDC guideline says that you can take off your mask if you're vaccinated. So don't just take off your mask, get vaccinated, and then you can feel comfortable to take off your mask. And that will eventually curb the new mutations.
Thanks. The next question is about post-COVID syndrome. Could you talk a little bit about that? And also, do other viruses cause similar long-term effects?
This is an interesting one. So, I don't know of a respiratory virus that will cause similar effects. Our ARDS is something that is really hard to recover from. It causes substantial damage that people need considerable rehabilitation if you're just talking about the amount of tissue that is damaged in the lungs. So that is what we are most kind of concerned with in terms of the long haulers of the COVID. There is a psychological side to it too, as we said, the anxiety that we are all living in, the things that we compromise, the fear, the different modalities of classes that we have to adapt to. So that is another aspect to it. But if I talk about viruses other than the respiratory viruses, yes, we've seen that. So once upon a time, HPV, we didn't know what was the consequence. What we later understood was that it was a virus capable of integrating and causing cancer. And so similar is, for example, HIV. So we just don't know yet what else is the virus doing. What is visible to us is that it is causing some, respiratory distress. Does the body fully recover? There is also, during the later stages of the disease, there is multiple organ failure, or multiple organs are affected because of that cytokine storm. Not because of the viral load, but because of the cytokine storm. So it'll be interesting. I mean, I'll admit I haven't seen or read too many of those papers on pathophysiology, but I can't imagine that there isn't an aspect of organ or tissue that is not damaged and needs to recover after COVID.
Thank you. The next question has to do with vaccine hesitancy. And certainly a common theme through so many of the presentations is the importance of the vaccine and getting to the point where maybe there's herd immunity, or at least getting closer to that. This question is really asking about where the vaccine hesitancy is coming from. Are there legitimate concerns? And I think with probably asking more specifically to COVID vaccines, not to past vaccines or history of vaccines, because there have been a lot of developments, of course, over the years. If you could say a little bit about, are there legitimate concerns that might lend themselves to being hesitant about taking the vaccine? And really, if not, how are we going to be able to get society past these unfounded hesitancy?
Yeah, so this is a difficult one, right? So this is something that I struggle with too. I mean, what I can say is that there are definitely age groups, and there's definitely medical conditions that there will be some adverse effects of the vaccine. What I can tell you is that vaccine development did not cut any corners, and there was no lack of testing of the vaccine. Now, what I can understand is that the scale at which we are vaccinating, we haven't done that before. We have recently expanded the age group of vaccination to 12. I'm keeping my fingers crossed before school opens, it's extended to, a smaller age groups so that my son can go to school vaccinated. But yes, if you hear about an issue in one of the kids, you're going to wonder, is it going to be my kid? Especially because, there's this aspect of COVID where there is a cytokine storm storm, and there have been some controversies, or not controversies, I guess, issues that have been raised for the AstraZeneca and the Johnson & Johnson. What I will tell you is the odds are, the data is true, but the odds are that you will have the number of adverse cases tell us that there is more benefits to getting the vaccine, and those population that are really should be concerned, or will show an adverse outcome, is very, very small, almost close to something that would happen without getting a vaccine. Now, what I'm not saying is that there is, what I want to stress is that there is a need to continuously monitor and collect data on which populations have these adverse effects. We definitely need that. If we can do something to make these vaccines better, we should continuously do that. But at the moment, it looks like vaccines are doing us more good than that. And so I really don't know how to get over vaccine hesitancy. What I know is look at the numbers, and all clinical trials are publicly available. If you need to look at the data, go by the data, not by the anecdotes.
Well, and if you don't mind, just as a quick follow-up, it seems like so much of that in terms of communicating that to broader populations is that don't just look at the vaccine and some possible side effects, but also contextualize it in terms of how would that compare to getting COVID, or what the risks might be with COVID.
Yeah, no, that's definitely one. And I think we do forget that the younger population is vulnerable. In fact, with the Delta variant, that's what I have heard a lot. And I've seen in India very closely with some friends and family, and that the younger population, the cytokine storm is much more larger than with the earlier variants. And so, yes, contextualize it, see it, in the light of what COVID would do to you rather than what vaccine. And if you are concerned, there are now choices, right? So I heard physicians say, if you have concerns about like the coagulation issues, don't get the Johnson & Johnson one. Now at least in the US, we have a choice as to which vaccine we can get. So talk to a doctor, talk to a physician, get the guidance. And as long as most of us get vaccinated, we'll be good.
Right. I think it's a relatively technical biology question that we have here. Do vaccines have to target the spikes of the COVID cell body, or could vaccines target other parts of the COVID cell?
Oh, this is such an interesting one. So this is something that I've always wondered. Okay, so the current vaccines are targeting the spikes, and that is because the spike protein seems to be like that outer thing that the virus first presents. It's so important for the binding. If you're thinking about the initial recognition, that is what the virus needs to do the first. So currently the vaccines that we have are all targeting the spike proteins. Now, should we make vaccine against other parts of the virus? Absolutely. Are they going to be equally effective? We don't know, but there should be research, because we should not put all our eggs in one basket, right? We should find other targets, because we should have things that have broader coverage, things that are not going to change from one variant to the other. So if the spike protein is the thing that is changing so rapidly in different variants, maybe we can have a combination vaccine so that we have one against the spike, which is truly effective, and then we have something against, something that says more consistent. So we have at least some protection against that. So currently all our vaccines target the spike proteins, but I hope someone, and I have to go dig the literature, is looking at other vaccine candidates on the COVID. I hope that answered, and I didn't take it in a different route.
Okay, so I have a question. It's actually a couple of questions, and they had to do with one of the readings that the students did. So if you didn't do your reading, pay attention, because you might get a nice answer here. So it had to do with a couple of some terminology. So one was the phrase waning immunity, and the other, let's see, I have to go back down in here because he asked it twice. And now it's, okay, yeah. Immune escape and waning immunity. Could you explain those terms?
Yeah, okay, so those are more sort of general terms. I mean, waning immunity basically means that when your body is, you get your vaccine and you're told that a month from getting your second dose, you're protected, right? How long? So this is what we were talking about. How soon after could we expect to need a booster dose? So eventually, fewer and fewer antibodies will be there, and your immune system will kind of need the waking up, right? Saying, hey, keep making those antibodies. And so waning immunity refers to that. The longer the time has passed since you've had a natural COVID infection, which has been shown to give you slightly less immunity than the vaccination, you can expect that your immune responses kind of go down, and you will need another booster, which is going to wake up your immune system and bring it back. What was the second one? Waning immunity and, sorry, I thought I had it in my
Help
head.
me, Anna, because I just erased it.
Oh,
immune escape. Connor's gone back on. Thank you, Connor.
Thank you. So immune escape basically means that your antibodies can no longer stop the virus, can no longer identify the virus, right? And that has to do with the change in this in the spike, which is what currently our vaccines are against. So you're making antibodies, but you're making it for the alpha variant when you have really now Delta variant, but the Delta variant is substantially different from what protection your current vaccine is providing. And so I skipped a slide, which was one of very preliminary studies which showed the effectiveness of 1 versus 2 doses, and it was comparing the Pfizer versus the AstraZeneca vaccine. And what it showed is that really after 2 doses, there is substantial amount of protection, even with the new variant, with the Delta variant. So that is some promise, right? And hopefully some motivation for people to get vaccinated. And what was striking was the difference between the first and the second dose. So what they saw was first dose, not so effective, but second dose really meant that there is a substantial amount of protection.
Thanks.
That was great. Thanks for that clarification. The next question, I think, touches on something you mentioned a couple of questions ago, and that is just really the phenomenon of living through this. We are all experiencing it, and we're learning a lot more about COVID than most of us ever learned about any other virus or any other pandemic, because we're living through it. And so this question has to do about the varied response. And we see that for COVID, some people have a very strong response and a very very deleterious effects, and some others it's a much lighter. And then similarly, is there anything to draw from the way the side effects play out with some of the vaccines, that some have stronger side effects than others? Is this out of the ordinary? Is this unusual to see that both for COVID and for the vaccine that we have such a variety of responses? And what do we draw from that?
Yeah, so again, I'm going to say that this virus is relatively new. We don't understand it enough. And it is still evolving. So there are a few things, right? So if it was the original Wuhan strain, then we would know much more, and our data would be much more consistent, and it would explain everything. Because the virus is evolving, and because the response from different age groups and people who have different underlying conditions is so different, that's why we see such different outcome. Going back to what I've been hearing back home, there have been so many more. I don't even know which wave they would call it now, but there's so many more individuals now who are in that age group 18 to 24 who have died of COVID. And that is because of the cytokine storm, because of the Delta variant. I will say that the data points that early intervention is the key. So if there's a very short period between the onset of the first symptoms to hospitalization in the case of the Delta variant, and that data data has showed up again and again. So how is this comparable to other viruses? I think we'll know in a year or two. We hope it doesn't change too much by then, but as of now, yes, we have seen a lot of variation, which makes management of the disease difficult. But vaccination and early intervention are two things I think that can help us in most of the cases.
Well, we're about at the end of the hour, but I want to ask you one more thing about the boosters. You know, we're a lot of us are anticipating getting a booster, you know, in the fall. Is the booster just to sort of provide some, you know, upkeep to the vaccination that you already got? Is it going to protect us potentially from new variants? Or, you know, what's your take on the role of the booster in the vaccination process?
Right. So this is where I think I ended my presentation, that, you know, we have to be ahead of the virus. And because the virus is changing, it wouldn't make sense for us to make the booster against the original Wuhan virus, right? I think it would make sense to even think of, and this is what, again, is the beauty of the mRNA vaccines, if it can be made affordable, you know, and I guess the government can subsidize and keep up with the vaccine rollout of the boosters also, is provide a cocktail that is going to protect you from the top 2 variants that are circulating, or provide you from a more broad spectrum, you know, thing that most coronaviruses will have, and something that is really a variant of concern. So I think we will definitely need boosters, that is for sure, because the way the pandemic has rolled out last spring, what did we think? We thought 1 week, and then we'll, like, you know, or 1 month, and then we'll come back. So it's not going away soon, but there is definitely reason to be cautiously optimistic, but only if we do our part, and only if we take our vaccine and then we take our booster and we keep the virus at bay. So it's going to be who wins the race, right? Could be neck to neck, and sometimes the virus gets a little ahead of us, and then ultimately the goal is that we go so far ahead that we don't give the virus a chance to mutate so that it becomes any worse.
Yeah, I think that's a great message to end on. So thank you so much for being here, Dr. Swati Agrawal. Appreciate your presentation, your technical presentation today. It was a really good addition to the class. And thank you all for joining us this afternoon.
Thank you.
We only have one class period left, and that is on Thursday, where our very own Professor Anand Rao is going to talk to us about COVID and how communication has changed. An important topic that we've heard about on and off throughout the course. And so he'll pull it all together for us here at the very end. So looking forward to seeing everybody on Thursday. Have a great evening, and we'll see you then.